several farms sign up, but we could only take 5 to participate so that they could be under the wing of Monique and Domingo and Daniel. And so we're very thankful to CT NOFA for their partnership with us on this project. And you'll get to see 3 of the farms today. RC&D, Sweetacre Farm, and Lathrop Farm are here. Or Common Ground and GBI are not able to join us, but there are examples of an urban farm participating in this project as well. So yeah, we're excited to hear the results. This project started off in June and it ran through September, and Mia and Amelia from UConn Extension have been very helpful in this project and trying to give it some scientific teeth, if you will. This is not something that's commonly being tested or done, and so we're kind of on the pioneer front of all of this. And so we're going to start off with some PowerPoints. We're going to move into a tour of the farm with Mia and Andrew, and then we're going to take a look at the components of the mobile unit, and then we're going to conclude with the farmer panel, and we'll have some food and refreshments and time to network. There's a porta potty on the other side of the garage here, and there's beverages at any time if you need something to drink. So with that, thanks, Monique.
Thank you. All right. Hi, everybody.
Hi.
So I'm going to be kind of doing this, but it's been such a thrill working with these farmers, and the enthusiasm around this work is really so encouraging. So I'm looking forward to Domingo and I sharing what we have found scientifically, and for me, not being a scientist, visually. We've got some microscopy to share with you that we've looked at. But I want to start with introducing Domingo, who really put all these test trials together. And you'll see the amount of work that he did in making that happen. So he's going to show the slides of his work and also the findings. So the plan was to compare compost tea. Does everyone know what compost tea is? Mm-hmm. All right, and we're going to be actually setting up a compost tea system here because we have this available compost tea unit, and so we're going to set up the brewer, and what you do is you can see I'm going to add amendments. I'm going to add worm castings or any other compost that you want. In this case, we're going to use worm castings. We're going to let it bubble for 24 hours, and we're going to do a foliar spray and root drench around our favorite crops. In this case, we took that compost tea as our compost tea side, and we also had a control side, and the control was just the amendments. So the amendments that we put in: fish hydrolysate, kelp, humic acid. And we compared those. And Domingo set up a very complex trial to prove the efficacy of compost tea. All right, so I'm going to let you take it from here. Oh yeah, I'll be the switcher.
Okay, my name is Domingo Medina. I own a small community composting business down in New Haven, Connecticut. I've been doing it for 11 years now, and my whole passion is to do high quality compost where the most abundant and rich and diversified amount of microbes, which I know are the ones that are responsible for soil health, and that's where my true passion is about. I have a stake on this project, in the sense that because I'm a composter, I want the project like this to be able to show and to be able to educate others of the importance of a compost. The important about compost extra and compost tea is that many times farmers cannot afford to be able to buy a lot of cubic yards to supply to their farms on a yearly basis. But what they can do is to basically produce compost extracts and compost teas that will allow it to use it as a way to, as a soil drench or as a foliage spraying, and expand at least the activity of microbes at the level of the root systems in the plants and also at the foliage level. So it's a tool, it's another tool that farmers in the organic world can have to be able to extend the value of compost in a lot of production. Right now, a lot of the people that work on compost concentrate mostly on the processing aspects of it. And the application is, I think, mostly concentrated on labs, on studies, concentrated around people that want to demonstrate the value of how we can capture carbon, how can we improve soil health, But even composting is not a regular type of recommendation when we talk about soil health. We talk about crop rotation, we talk about no-tilling, we talk about other practices. But compost is mentioned, but it's not something that people take truly in-depth understanding of the benefits of this. So anyhow, so as Monique explained, we just wanted to introduce to farmers here in Connecticut the value of the concept of compost deepring as as a set of tools to be able to incorporate into the daily practice. The idea is to eventually move away from organic or inorganic fertilizers. The sense here is that a lot of our soils have enough nutrients in the soil, and what it takes is a good, healthy ecosystem of microbes to be able to provide for the nutrient cycle. Okay, and there's a whole debate about whether you amend or not to amend. But the idea that people that are into this field is, as you keep up the biology in place and healthy and happy and healthy, you're going to have that access to nutrients that plants will otherwise will not be able to access, given that they don't have the capability. They need that symbiotic relationship with microbes in the soil. So we went out to do experimental trial. We did it in 5 farms, and we wanted to, by doing this type of application, we wanted to increase the quantity and diversity of microbiology in the soil and the plant. We wanted to optimize the nutrient cycling. Hopefully, we wanted to be able to see what was happening at the level of the leaves of the through tissue analysis. We wanted to look at would this application have an effect on pest and disease incidents, and overall, can we expect some crop yield that has been enhanced in terms of its quality. So this is what I was mentioning. The compost tea world, there's a lot of benefits, but there's a lot of unknown. We as composters, we use microscopy to be able to look at the abundance and diversity, and we do it through morphology. And we do it through morphology, try to recognize the presence of bacteria and fungi and protozoa and nematodes, all those beneficial microbes that we know play a role in the health of the soil through all that food web that guarantees that nutrient cycle. Okay, but we do not know specifically who are they. Who are those that help in the nutrient cycling? Those microbes that are into the disease suppression, those that are decomposers, those that are help in the plant growth promotion. This, and of course now in the use of health and bioremediation, we don't know who they are, and now we're moving towards an era of DNA analysis, where people are starting to understand who are these microbes and what are the roles in the soil for all this application. So somehow, as a composters, we only go about trying to provide as many and diverse amount of feedstock, so we can provide food for a wide array of microbes to optimize, hopefully, the effect of that they can bring to the soil. But we're kind of blinded here. So that's why each time you do composting, the diversity of feedstock into your compost mix or your recipe goes towards that, trying to enhance that population of microbes. And so this is a new era that we're starting to see, and there's a lot of results that are coming out, mostly from labs. But then the idea with this is to fine tune. If I really want to tackle, early blight, or really want to tackle any kind of diseases, see who of these bacteria can play a role of competition or suppression, and that requires, a lot of experimental work. Anyhow, we went through a process, and it's a process that required a trial. When I was invited to participate, the trial was making that we have to organize this type of study through, you can't see it here, but we started with, establishing, our objectives and our research questions. We were able to identify what are those variables that we wanted to work with. We need to choose experimental design that can guarantee that it's, the things are different and not because, it's just an anecdotal observation. We had to look at, choosing the field and the plot and mapping it and organizing it. We had to go and do our applications and harvesting to the analysis of the data, and then to draw conclusions and to share that with the public. So we went through that process because if you really want to demonstrate that in work and you want it to be adopted by farmers, somehow you want to take out the human bias. Again, as me as a stakeholder, I want to see positive results because that's what I do. But I also need to adhere to certain standards when it comes to things like that. Next, please. These are some of the research questions that we already mentioned. Next. And basically we did a Compost tea application. We did what's called a pair comparison design, where we did a minimum of 4 replications. So 4 plots assigned for control and 4 plots assigned for tea, and they were randomly chosen, okay, with certain arrangement, which I'm going to explain later in the. But we selected the farm. They were invited to participate. They committed to the type of requirements that NOFA was providing for this experiment. We established a baseline, basically information about water, groundwater, water that would be used for irrigation. We looked at soil. We not only looked at the pool, but also we looked at what was soluble, what was already available, and we looked at it from the beginning and at the end of the trial. We looked at, of course, each farm had the liberty to select the crop, and of course the particular land that they wanted to set aside for. And then we went out to do each plot received 5 photo applications spread in 3 weeks. So every 3 weeks we were going around preparing some of the tea beforehand, then applying the next day. A lot of traveling, but that's what it required. And then we concentrated around yield. Yield is something that was measurable, that we could capture, that it doesn't want too away from what farmers do already in terms of they need to collect their crops. So the adding effort here was to tally and to capture that pound. We also looked at some of the observational plant and crop diseases. We did not do it systematically. We did by the observation of the farmers. So we were waiting for the farmers' feedback in terms of their perception of the overall health of the crop during the season. From the very beginning, we looked at compaction of the soil. We looked at anything that possibly can affect the viability of microbes in the soil. So all the farms were okay with compaction. Soil structure was also it varied. We had farms that had 8% organic matter, and others had 12 or 15. And the difference is that you have farms that have been cultivating that piece of plot for 12 years, where others probably have 3 years or 4. So there's a difference also on the history of the build-up of the soil for this. And of course, we did a lot of the soil biological assessment before and after, and we also did it for when we did our tea, making sure that the microbial population that we saw in the vermicompost somehow was in the solution of the tea in a relatively same proportion. And there's a correlation in that. And then, of course, we were looking at plant nutrient uptake. You were also limited in funding, so we only did it once at the mid of the season, just to make sure that plants were uptaking the nutrients they needed to be able to optimize the production of the crop. And then water quality. Water quality, we had this I think is a variable that played a role. In, the Common Ground High School and Reservoir both have used city water, while ARCSA, Mendy, Sweet Acres, and Lathrop use well water. And I think that's an important variable because we don't know how chemicals that are done to be able to purify water in the city has an impact on the microbes. Next. So this is an example of Common Ground High School. This is they set aside 2 large rows for growing kale, and that's Diane there. That was very important in this trial. Sorry you can't see that, but this is the early stages of the crop. Next. And this is an example of the, while we were doing, one thing of the challenges of this type of research is that because we're doing foliar spray, we might, you might have wind, okay, or you have proximity with the other plot that may have an impact. That was a concern. So we were using these panels to try to separate and minimize as much as we can. Okay? In a perfect world, you have nice plots well separated with a barrier to avoid that, but there's always a concern of how you minimize the impact. You probably have an edge effect, and we're hoping that that wasn't relevant in all the study. Next. And this is reservoir. Reservoir had a lot of crops. Reservoir, sadly, Diego had a family that died early in the project, so it was hard for him to attend the project, and depending on decentralizing this effort among a lot of people. But still, they had results that we were able to observe in the field. Next. Then we have Lathrop. They work with 3 varieties. They work with cucumber, they work with tomatoes, and with watermelon, big watermelon, heavy watermelons. The watermelons are not good for this type of trial because they just run through the floor, and then you can't identify which plant is which. So we stopped, providing them with tea, at least in a differential way. Next. And of course, this is Andrew pouring some tea on me. I don't know why. I think he feels that she needs to grow a little bit more. Okay. Next.
She's wearing a backpack sprayer, just to clarify. She's wearing a backpack sprayer.
Oh, okay. I didn't see it. From the angle, I didn't see it.
But not a drop of tea, because we had 50 gallons,
Yeah.
right? We always had left over. Not a drop of tea was wasted.
Yeah.
You planted.
I thought it was a little bit mean, but now you clarify. Thank you. Okay. This 3 acres. 3 acres was able to establish 2 varieties of tomatoes that we were able to test in the trial. As you can see, we had farms that used a greenhouse, another farm that were openly in the field, another that was the first time the farmer growing tomatoes in a new field. So with great variety of experience of the farmers and the techniques to grow food. Keep going. This is important because when we're going to see all these pounds per acre, pounds per square feet, sorry, you have to pay attention also that different method might draw different type of results also. It's just an open question in this regard. And this Arsamendi, this was a very interesting plot. She did a lot of intercropping. She included a lot of companion plants. She had a blast, basically. So I wanted to respect what each farmer wanted to do in terms of how they dealt with weeds, how they dealt with diseases, how they dealt with how they grow their crops. Next.
This is the first year, by the way, that this garden exists.
Okay, so this one more or less looks like full replica design. This is Common Ground High School, and they had these beds that were, each plot was 8.7 by 3, and what we did, because of the issue of potential contamination, we always selected the first one at random, and then we alternated. We wanted to have 2 plots continuous of tea, 2 plots continuous of control, to ease the pain of us going around spraying, but also to control a little bit of that factor of cross-contamination, if you can call it that way. Okay, so these are 4 blocks, and basically we were comparing the mean of these 4 plots with the mean of these other 4 plots for control. The same here with some of them misinterpreted, what we were trying to say. So they only were able to do 2 blocks for a variety. So we only have descriptive statistics for these, but not more meaningful statistical analysis. Okay, next. And this is we were very keen to understand and map out and to understand the soil conditions where we where we're looking at in terms of our trial. Next one. And this is example of 3 acres, the same thing. We randomly selected the first plot, and we went from there. 3 acres did have another row here that separated this group from this group. So that's the only one, but we needed to provide divisions each time we were spraying. And that was challenging because they were okay when they were up to our height, but then they grew well above, and that was at the end a little bit of a challenge. Next, and this is the mapping. Next, and this Lathrop. These were the 3 crops I was telling you. You know, we worked with particularly with the cucumbers and with the big beef variety, and then you had the ones with the watermelon, the sugar baby. Next, and you can't see here very well, but that was part of the design. And this RC&D. RC&D tried working with 4 blocks of the big beef and 4 blocks of the fairy tale. They work with eggplants and so on. One thing to mention, next. One thing to mention is that sometimes teas are not effective if you're already starting with a diseased plant. And we had the example here of Lathrop that they had an issue of early blight, even coming from already from the greenhouse. And we were puzzled, well, where is it coming from? Is it coming from the compost? Is it coming from, from the growing media? Where is it coming from? So now the concern is how do we guarantee the seeds are well protected from the beginning? Whether you apply heat, whether you drench them with compost teas and inoculate them from the very beginning, and that also happened with the cucumbers. So so for us it was very difficult to catch up with that spread of disease coming every 3 weeks. So these are some examples of the soil, all the data that we were collecting. One thing that we're very keen about, and it's an innovation here for us, is that somehow we needed a number to be able to discern whether our tea production was of quality. And the thing with microscopy is that even 2 experts in microscopy looking at the same sample may come with a different result in terms of what they observe. So it's very hard. So we went on the qualitative way to be able to observe those the existence of those beneficial microbes that we wanted to see, and we wanted to see there was all the trophic level present as much as possible. But then we started using a tool that's called a microbiometer that measures somehow the microbial biomass and even the fungal to bacteria ratio. Okay, and it's been controversial because there are people that believe it, some are not, but they seem to have done a lot of trials where they correlate their finding with the observation in microscopy. So I had to say, let's use it, because if this goes in the same direction of what we're observing, then we can use that as part of the learning curve. It might not be the explanation, but again, you're using a lot of tool to observe is what you're doing is leaning towards an impact that you want to see. Okay, and you'll see some of those numbers. We went and looked at tissue analysis, the yields, and these observations by the farmers of the disease. Okay, next. Okay, this is just a calendar to show, every 3 weeks we were showing up and preparing. So we were doing some brewing in Common Ground High School, and the brewing was done here in Lathrop. We wanted to minimize the movement of brewers, between the board, the east and the west of the state, and to make sure that things were ready by when we arrived. We were short-handed in terms of how much time we could spend. So it was very convenient for us. We prepared all this around our convenience in terms of when we could come and spray. And these are the data collection, things that we were doing. This was able for us to be able to understand our data collection. Next. Okay. One thing that, given that we had 2 different sites, we actually did 2 types of 50 gallons, of 2 types of brewers. One is the 50-gallon that we have here, and the one in Common Ground is 7-gallon. And we made sure that the ratio of compost to water was kept properly. So, we were not doing any dilution that was differential in that regard. Next. One thing that is critical to understand also is that when you provide, we were providing humic acid, fish hydrosalate, and kelp to our recipe for our teas. And when you're training this type of technology, you know, they emphasize that humic acid is a fungal food. The fish oil salate is for fungal food, and the kelp is for bacterial food. But what they don't emphasize in the training is that each of these ingredients or amendments also bring their own nutrients, you know, like, you know, nitrogen and sulfur and phosphorus and calcium and zinc and iron. So you're providing also food for the plant along the way, and that somehow needs to be paid attention to. Here is how can we make this? How can we discern the effect from the actual activity of the microbes in place? Next. And this is some of the directions that Monique gave us in terms of preparing the tea. That we're going to go over it later on. Next. And this is another thing that we corrected, by the way, Amanda, that we had the discussion, is that you know, not only the compost to water ratio is important, but also are you adding the same amount of food for your 50 gallon, for your 7 gallon, and for your backpack or 4 gallon that we were using for the control. And what I realized is that they're quite the same. So we were concerned that we were adding more food to the control, but I was doing the math wrong. And Monique told me, you're not
As a
looking well.
mathematician.
Yeah. Okay, next. Okay, and this just to show, using the microbiometer, that we were able to look at the biomass and classify the quality of the tea. So these are the first brewing, second brewing, third brewing, you know, that was changing over time. And we were able to measure with the microbiometer that biomass. And the same tool will give you a fungal to bacterial ratio, which are pretty high for what I tend to find in a regular compost. But you know, we're dealing with vermicompost. And you have to test a lot of vermicompost to see what's something that it's that is standard or that has been surveyed. The microbiometers show you some of their own standard, but it all depends on the sampling and how frequently, you know, and making sure and control for the feedstock that have been given to vermicompost and so on. So in our case, you know, we had, you know, application. I don't know, I missed some data from the beginning, but you know, we were going between above average and excellent in most of the time. And then in places we had below average, we went out and see that we had issues. There's a correlation, high correlation between the microbiomass and temperature, okay? And it's significant. And what happened is that, you know, we had days here that that water that's coming from a well was 55 degrees. And we were trying to look at the micros one, you know, what's going on? And that's when we were instructed by Monique that we should be filling out the tank the day before to allow for the water to warm up to guarantee better conditions. And I think that was very important to look at is this dissolved oxygen. As you feed the microbes with this food, they grow and they multiply, but also they're consuming oxygen. So if the oxygen levels are not that good, it can become anaerobic, okay? Or you end up killing, you know, a lot of microbes, the ones that you don't want to select for those anaerobic microbes. So we are paying attention to that, measuring, and they were all optimal in our regard. And there's high correlation also we saw into the quality of the compost and the tea. So somehow our technology was transferring that microbial mass to the tea in the proportion, of course, because we are diluting along the way. So that was positive. Next. Okay, let's talk about results. This is a table that's going to be difficult for you to see from above. But what you see is the farms at the top. You see the treatment of the control and tea. You have the square footage assigned for those series of plots. You have the crops. Then you have the number of plants that farmers put into those plots in aggregate, of course. The number of harvests. How many times the farmer went out there and looked and harvested and wrote down that data. We looked at also the pounds, the yield per pound, and the pounds per square feet. The result is that the yield was relatively higher in 4 out of 7 tomato trials, in the sense of for the tea, okay? In 4 out of 7. Yet, inside with 4 replicas, The difference between control and T with respect to the dependent variable yield was statistically significant in only one of the 5. Okay? Even though we have some direction in terms of more production with the T application, it was not significant only just in one, and it was in here. You know, and I was surprised. Why Lathrop, with all the struggle that they had with those plants, they were, I mean, you're gonna see the graphic next. So this is, what you wanna see is this element here. Each of these points are averages of the control points that we collected over time. And you can see that they don't overlap in terms of the spread, in terms of the standard deviation. So they separate very clearly in terms of the yield that the control hole and the T had. Of course, we lost a couple of plants, but you recover them. How that impacted, I don't think so, because it's pretty clear how much of that happened. This was the only strong indication of that the T was working. Next. And then I took all the tomatoes of all the farms and put it together in one database. And the T came out a little bit higher in terms of average, but you can see the distribution are overlapping. Mm-hmm. So we can't claim any clear impact at all. Next. And this is the yield for kale. Common Ground High School was producing kale, and Reservoir also producing kale. And again here, the yield was relatively higher. For T treatment, 3 out of 6, so 50-50. And the difference between control and T with respect to yield was not statistically significant. Mm-hmm. Something about the kale, I know that kale and the brassicas most of the time don't have mycorrhizal relationships. And so that's a whole thing to research. You know, we didn't think much about this. We just allowed for the farmer to take it. But something about this thick leaves of the brassica, when we were pouring, you know, the whole tea just run away. I'm not going to take it, basically. So I have all my protection already, and that was so we ended up, you know, doing a lot of soil drenching with them. Next, and this is one example of the curly of the field where you have, you know, the control pretty high. This is something that I want to check with Common Ground, because something that I was observing is that I wasn't clear enough with the farmers that when you harvest, you have to harvest the same block, one block. Okay, that means you have a control and a T that is a block, and you have to harvest them at the same time. Otherwise, you're missing data. So you had, I forgot to mention that, you have some crops that were harvested more on the side of the control, and some that were more harvested. And it was convenient. Listen, we need to go to the market. We have to pick it up, or you send somebody else. So that's a factor to play into. Next. And this is the cucumber. Again, it was leaning towards T, but you know, the season was short-lived because of the challenges with the aspect nose here at the farm with the cucumber. Next. And this is the data that doesn't show any significance at all. But I always like to show the distribution of points. Next. Okay, another result that are not completely conclusive, but we also observed the data, is that we certainly saw an increase of microbial biomass from all the sites from the previous to the beginning. There were clearly an increase of microbial biomass, and we saw it not only with the microbiometer, but we also saw it with a microscope. And you would say, oh, okay, listen, that happens normally. If you don't have any plants growing on the soil, and then you put a plant, then you have all this microbial activity going on. And you should expect an increment. But we saw, at least on the 3 farms that were on this side of Connecticut that was using well water, the percentage of change of microbial biomass was much higher compared to the control. It didn't happen with the others in Common Ground and Reservoir. Was the water? I don't know, but that's something passing to. Then the other thing, data that I was discussing with some of you, there's a clearly increase in the percentage of organic matter on the soil. I mean, I didn't put it here, but it was staggering. Can this possibly happen from you know, a 6-month period? And then we were able to see some changes in soil irrigation that we're going to see right now, where the aggregates there was a clearly observational change of aggregates in the soil.
Can I just say something?
Yes.
These are micro aggregates. So what they are, bacteria being sticky and the glues from the plant will cause them to form these micro aggregates, which you'll see when we look under the microscope are sort of odd shaped or naturally shaped copper colored elements that hold on to moisture, that hold on to nutrients. So they're really important to have in your soil, and the microbes are what are creating them.
Okay, next. So just going to show you this graphic here. So you can see this is reservoir, control and pre and post, and you can see there's a change. They all increase in microbial biomass. You know, but when you look at the percentage of change, you can see that Arza, Sweet Acres, and Latha has a very high percentage of change from the previous to the next. And so for me, it's positive, you know, that that's a clear difference, and that I would like to keep exploring in terms of the impact of the tea. Next, and then we have. You can explain this to you. You already go ahead.
Right. So this is the microscopy I did. So Domingo collected samples of the control soil from the control side and from the compost tea side. Looked at it under the microscope. It's hard to see on the screen. They are copper colored, you gotta believe me. But we're at 40 times magnification, so it's a wide angle view on a microscope. How the density, so you can see all those spots and how much thicker it is on the bottom right. Those are the aggregates that if you zoomed in, and I have a zoomed in version, are much greater. And so I had also done microscopy at the start of the trial. So I just did it for this one to compare. So for example, on top is the control, before and after trial. It's hard to tell because the lighting is different, but they were somewhat the same. But at the bottom, you can see, again, the tiny little dots versus the more established, larger aggregates. And so these are, I'm just gonna go through these quickly. Arza, you can see the control versus compost tea. This one is now 400 times magnification. We're still at Arza here. You can see those aggregates, right? And actually in this one, I also found a nematode, which is a predator. I take a picture of that. And this is common ground. I found a significant difference in terms of microbial or soil structure, if you will, whatever you want to call it, microaggregates.
So when we talk about the soil structure, the benefit of applying a compost to your soil is one of these effects, the aggregation of organic matter, which helps in water retention. It helps in other effects in terms of controlling for temperature, creating a viable environment for microbes to live in, and these are the kind of things we like to see as one effect. Next.
And that one had less, and we did interviews with the farmers, and I don't want to, we're going to be interviewing them again here, but one of the funniest lines, if you don't mind me saying this, is, okay, Sweet Acres, 12 years of working on the soil and making this amazing soil. And so are we really going to make a difference if we come in with sprays once every 3 weeks on the 1 year? Arza took land that had never been grown, cleared it, started the farm, started planting. And so we found much more difference. But certainly in terms of the microbial activity and the formation of aggregates. And Liz said, well, if you give me 12 years, then we'll see what. That was so cute. You don't mind me sharing that?
I don't at all.
Okay, excellent.
It's true.
It is true. So anyway, if that doesn't come up, at least you heard it. And we do have, and this is it zoomed in, and we found, more fungal hyphae, just a totally different feel, if you will. But I just, okay, I'm gonna stop talking.
Okay. So these are the effects that you want to be able to see: the build-up of organic matter, the increase in microbial biomass, and this may not happen in a season. It might take 2 or 3, as we feel that farmers that have 12% or more organic matter in the soil take advantage of. And compost is one way with other soil health practices that allow to maintain those ecosystems, soil ecosystem viable and productive. This is just the one plant tissue analysis that we did. The colors, this green color tells you normal parameters according to Penn State University. That's when I send basically all the reports. They didn't show any deficiency of nutrients other than Common Ground High School with copper in their kale, and Lathrop with a deficiency in zinc for the tomatoes. The rest, all these plants, all this measurement that we did, what tells us that we were dealing with healthy plants overall. And sometimes we look at a plant and we look that they have some disease or some pest, but they still have the capability of being productive in terms, this was a concern of us. And of course, because we don't have a pre and post type of analysis, it was hard to say, but at least for me, all the plants we were dealing with were healthy plants from the nutritional point of view. Next. Lessons learned. So we understood as the season went by, as we got more and more of our prepared tea, is that tea preparation and quality, consistency. You need consistency, and that parts from bringing a high quality compost that has abundant and diverse amount of microbes, and sometimes there's a whole movement now in California where people look for this, prime type of compost because of that great value of it, and they're selling it for $700 a cubic yard, okay? And of course they're selling it to orchards, and they're selling it to vineyards, and they're selling it. So what farmers are doing is that they're moving towards doing compost extras and tea application because those prices are pretty high. But that's a little bit tell you where the market going. So consistency is something important. So the controlling of the temperature, to have good microbial biomass along the way, these are parameters that look like difficult to control for, but once you have a good recipe and you have done several measures, it's like cooking your same pasta every Thursday night because you know it's going to taste good and things are going to come out nicely. So there's a learning curve here of understanding your inputs and the impacts of your inputs. And of course here will be, understanding who are the actors here, who are the microbes, what type of role they might bring in through the application of the tea, as we can identify them and see what role they have. The time of application, the idea was this, to do it in the early morning or late evening when temperatures were cooler and the amount of evaporation was lower. But at times, we only were able to come on at noon time. We hated it, and we were at 3 o'clock in the afternoon, and the heat was killing us, and Mia was, dead already. So, the tea application is very important to pay when to apply it. Also, to pay attention to apply not at your convenience, but at the stages of growth of the plant. So, at the seeding time, at the transplanting time, at the early growth stages, at the flowering time, at the fruiting time, at the beginning. That will be probably much more supportive along the way of where the plant is going through transition and is less vulnerable to the present pests and diseases at the time. It was convenient for us, but probably if I would do it like this again, I wouldn't run around as much and concentrate it in cases that we can see much more clearly. What else? The application according to plant growth, I said that, and this is a description of how to do it. The number of harvest events is important, and I should have emphasized that, and I didn't do it enough, is to harvest per block and not per plot, to make sure that we always had a comparison of the control of the tea over time. It didn't happen to all the farms. It didn't happen with Arza. It didn't happen with Sweet Acres, and I think it didn't happen with Lathrop. But it did happen with Common Ground and with Reservoir. And they were working with kids, they were working with summer programs, they were working, so this probably that didn't allow for the full attention to the importance of paying to details when you were coming to harvest. Then the length of the harvest season, we stopped early September, but you still have a growing season. And we didn't take, we can't say if this was a trend to continue and things that could have been different story in terms of the results, but the length of the season is something important to truly have an understanding of your yield per acre. We only say this is your yield for square feet, from end of May to the 1st of September. Beyond that, we don't know. Then the other thing is that the importance of the initial plant diseases to minimize and control it as much as possible. This issue of the potential drift or migration effect that we were concerned about. When we're doing the trial, if you're not doing a trial, you're spraying, that's not an issue. But if you want to demonstrate changes, somehow you want to minimize that by physically separating the plots, putting some barriers like we tried to do. Is a factor. We don't know if it's truly affected or not. We won't be able to say. But and then of course the farm management practices is something that's important. That whether you're controlling a disease and putting copper, if you are controlling a pest, or you're doing intercropping or cover or companion plants, or you're doing a greenhouse, each of them has a role to play, and that's very important. And then the last, this is something that you want to show.
Yeah, I got these beautiful photos that Nia sent, and apparently they were delicious, and it was the first time they had ever produced fruit. And the only difference they did this year, guess what they did with the leftover compost tea? They applied it to the peaches, and I do that at home, and I can't eat all the peaches either. But amazing results, and you also, and I don't want to step on what you're going to say.
It was the first time we had fruit on those trees, and it was hundreds of pounds of peaches. The first time ever. And I'm
Mature
convinced-
trees, like they were ready, they should have been through.
Yeah, they're right there. I'll show you the
Yeah.
later, just a handful.
Crazy.
Very sweet, very sweet.
Okay, so that's our presentation.
After all the farms were sprayed, we got the benefit of the leftover tea. And I think we had 2 hours, Monique, I don't know where she is, 2 hours to apply it. And so we applied it to our eggplants, our peppers, summer squash, our herbs, our tomatoes are back there. Did not spray the tomatoes that we weren't supposed to spray, but, and then, and like we said, the peach trees over here. And it was, to me, it was really significant, the vigor for all of the, crops that we had. Although we had challenges with the tomatoes and the cucumbers, we have those challenges every year. And but all the other crops did amazingly well. We haven't had summer squash in 2 years because of the disease and pest problems. This year, we just kept producing and producing for, the entire season, just till October 7th.
And also I've noticed that as there's a lot of turnover that needs to happen here for before the winter, but as I've been pulling out some of the root systems on a number of the different plants, the escarole over there and lettuce, there are way more worms in the soil than I've ever seen here. So whatever we're feeding from that tea, I'm certain it's the soil's benefited.
We could just take a walk
The
this way, just
microbiome,
stretch
yeah.
it.
To say how much of these amendments and castings you need for a 5-gallon bucket. But we are doing, usually we do 50, but it's end of season and we're not giving it to any other farm, so there's 30 gallons in here. So maybe you can help me with the math, but generally, I got this, here we are. So if it's usually a cup for 50 gallons, how much would it be for 30 gallons? Thank you. 3/5, okay. Really, I can do this. Can someone help me? Because I've got, yeah, that would be helpful, thank you.
So we're gonna have to get that one.
I know, right? 3/5, okay, if I'm gonna do a little more than a half, how about that? I can't even see the half measurement. Oh, I think I got it, okay. So there's that. And then I always have, that was kelp. This is, yeah, it's just easier to measure and use. So fish hydrolysis, I like it so much more than fish emulsion. It's much more nutritious. It's a cold process, and it's actually, they keep the oil in it,
with fish emulsion, they heat it, they lose a lot of the nutrients, and then they sell the oil separately. So forget it, right? So fish hydrolysate, and then I usually do the ratio 4:1, 4 parts kelp to 1 part fish hydrolysate. So this is really eyeballing it. But how bad can it be, right, if you add too much fish? What are we at there? A little bit above half. All right, so I have a little bit more. Yeah, it's coming out weird, so that's why it's hard to measure.
Monique, any reason why you guys chose every 3 weeks rather than weekly or?
I just have that ritual,
Yeah.
what I do.
Yeah.
And there's something else, talk about going into way too much detail, but what I'll tend to do at the beginning of the season, I'm thinking we want to put in as many microbes as possible. So I will take castings right below where they're feeding because I know where that's the most microbial activity and all the predators and everything else. Oh, I'm shit. I got my hand up. But near the end of the season, all those plants want is nutrients for their seed, their fruits and things like that. So I'll tend to get it further down when the castings are more mature and less. What do you think about that plant, Mia?
I think that sounds great.
Okay, cool. This is a splash. Okay, this is my idea of a splash of humic acid, which is a really good binder and has other nutrients. Okay, so I'll take this. And I'll throw it in. All right, so those are the amendments. And then it's just a simple process. If you only have chlorinated water, you're worried about chlorine, I put the amendments in and let it bubble. You're fine. The humic acid helps to neutralize chlorine. All right, so this is 10 pounds for 7 gallons. So instead of 10 pounds, I'm going to do a little bit more than half. And this is, oh, there's some worms. No kidding, worm castings. All right, so we put it in there. Dump in about half. And does anyone have any string? Because I realize string. I want to dangle it. Usually you can just have it hooked on the surface.
Just gonna go get some tomato twine.
Okay, awesome. And so the other key here, which we always try to do, if you want to come around and see me do silly stuff.
She's got a
And then what I'm doing, I'm massaging the castings and getting rid of aggregates and stuff. And I'm releasing the microbes into the liquid so that they can start feeding on those amendments. So they're going to have a party for 24 hours.
Why is it 24 hours? I guess I never asked that.
Well, from what I learned under Dr. Lane Ingham, who had done a lot of experiments, and she seemed to think that was when the microbes had multiplied the most, had the most nutrients taken in, and weren't getting bored, and needed to now go into plants to start the symbiotic relationship with the plants. So that seems to be optimal. We, sometimes at organic nurseries, they'll brew it over the weekend, and I've checked it under the microscope. Everything's still living. It has a 100-foot hose, so you can sort of park it and then spray with the long hose. The other thing, if you attach it to and I'm not
PTO.
A PTO, it has a vibrate. What do you call it? Vibrator. When it vibrates. Agitator of some sort.
Well, the PTO pressurizes the tank.
That's right. What he said. Do you want to explain this? Because this is beyond my capability. But it also has a really good strainer, so it never clogs. And they put it onto a small little tiny tractor. And I'll actually I wanted to include a photo. I'll include the photo of how they set it up. And they'll take a whole thing, 50 gallons, fill up the sprayer and go into the field and just spray everything. And last year everyone had those terrible, tomato wilt or downy mildew. Everyone lost their tomatoes around them. everything was perfect with their farm. So it eats powdery mildew, downy mildew, botrytis, rust. So